This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1245, recorded on August 15th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And here in Western Mass, it is the first non-scorching day of the week, and maybe the last. It's 83 Fahrenheit, 28 C, moderate humidity. Very nice.
Just realized, Alan, you're all white now, aren't you? Your hair.
Well, I was born that way.
Oh, your hair.
Oh, my, yes.
I wouldn't say something like that. Your hair and your beard are all white, right?
Yes. Yeah, they've gone pretty much completely
How
silver.
old are you, by the way? Are you 50?
I'm in my 50s.
Yeah, that's not bad. I just read a really... Let me introduce from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is 87 Fahrenheit, 31 C here. It's pretty warm on the humid side, though not the worst we've had this summer.
See what it's here in Chelsea, 31 C and sunny. Yeah, it's not bad. It's been very nice weather. I like warm weather. I don't like 100 weather as much, but I can tolerate it. But it's no longer 100. I was just going to say, I read a really nice article in it was in New Yorker. And it's about longevity research. It's very good. All the billionaires in this country want to live forever. It's really weird.
I was interviewing somebody recently about longevity research, and she said, we've discovered a new model organism for studying longevity. Billionaires.
That's right. They have these clubs that they go to, and you pay a lot of money. They give you supplements. They check everything every day and make you do this and that. And they actually talked to some scientists, also the Buck Institute, where Eric Verdin is the head. He used to do HIV, is involved in this. And one of them said, basically, one of the scientists said, if you're interested in improving your health, that's great. But improving your lifespan is BS. Although if you improve your health, you will improve your lifespan.
The way I've heard it framed that I think makes the most sense is, everybody's going to die probably in less than a century from their birth. And really what you care about most or should care about is not how long you live, but how healthy you are for that time, because that's how much you can actually enjoy life. And so he was referring to health span instead of lifespan.
Health span, that's it.
Maybe the better way to frame things.
That's what this guy said. If you want to improve your health span, it's great. If you want to improve your lifespan, it's BS.
Yeah.
Yeah.
And as anybody who reads science fiction or fantasy should realize, immortality is a horrible, horrible idea.
Yes.
Yeah, just ask any vampire.
Any vampire, any buddy who's got some time travel curse or whatever. Yeah.
It's a good article. They had a lot of things that correlate with long age. And I can't remember a single one now. But I'm going to bring the article next week and read some of them.
Well, the specific interventions that keep coming up are exercise.
Yes.
Eating a reasonable diet that's not all ultra processed fat and sugar.
Right.
And getting adequate sleep and being socially active are recurring things. And then there are some drugs that are being investigated, but really, it's a lot of free stuff that people can do.
It's a lot of money in this.
Yeah.
Huge amount of money.
Yeah.
I mean, some of these people take 80 supplements a day. It's like, what are you doing to your liver and kidneys, right? I mean,
and also, if it's about living longer, healthier life so that you can enjoy yourself, how much enjoyment do you get about taking 80 pills a day?
That was my thought, is that at some point your life becomes about taking supplements, and that's not very enjoyable.
No.
I can't even remember to take one pill a day sometimes. Oh my gosh. Because you have to go get the pill, and that's the problem. If you have 80 of them, they're going to reside in a container, and you have to remember where you put it. Anyway, it's a fun article. I didn't make it a pick because the New Yorker is paywalled. But we subscribe here at the incubator to the New Yorker and the Atlantic. And so I thought we should have some reading material, like a doctor's office.
Yes.
If you enjoy these conversations about rambling things, actually, we do talk about science here. But sometimes I have an idea in the beginning and I just want to talk about it. We'd love your support. Microbe.tv/contribute. We give you science, real science, no lies, no disinformation. We try and explain it all to you. And we need your support to do these programs. We have 9 different programs, right? And we would love to have your support. Microbe.tv/contribute. This Microbe TV is a 501c3 nonprofit entity, so your donations are U.S. federal tax deductible. There's a meeting, an online meeting that starts tomorrow. No, it starts Monday. So maybe a few people will be able to see it. Anyway, they asked me to tell you about it. This is from Amy Kistler, who is at the Chan Zuckerberg Biohub. The Chan Zuckerberg Biohub. Dear friends at TWiV, on behalf of myself and co-organizers Carolina Arias and Noam Stern-Ginosar, I'd like to share news of the upcoming symposium, Frontiers in Comparative Systems Virology. We've assembled a fantastic lineup of speakers covering diverse topics in comparative systems virology. Symposium will be held Monday, August 18th and 19th. And it will focus on strategies to sustain our community, build collaborations, and develop rigorous cutting-edge comparative systems virology at local, regional, national, and international levels. If anyone at TWiV is interested in learning more or joining online, please reach out. We also think the output from our Day 2 discussions on fostering and supporting our community could make for an interesting discussion topic for the TWiV audience. Feel free to contact us. So there's a website. You could go register. So I'll put a link to that. I registered. I'm going to sit in on it.
I'm looking at the list of talks now.
Nels is there.
They look really interesting. Yeah, I see Nels is there.
I think they should have included a podcast because I think a panel discussion is actually more interesting than someone getting up and showing slides. Right? Where you could have 3 people showing slides and then everyone's going to start dispatching, right?
Mhm.
Yes.
So you have a panel discussion. That's the way it happens, right? Okay, we have some news items. One is from Alan Dove.
Yes. So this is an article in Science. Let's see, where did I put it? Here it is. Reported by Kathleen O'Grady, and it's covering a paper that came out, Proceedings of National Academy of Sciences, and this is reporting a study by data researchers looking at publication patterns in the scientific literature. And what they discover is that there are patterns, very telling patterns of publications and authors, and especially editors and journals, cranking out fraudulent papers and doing it at scale. This has actually become quite an industry. It's especially prevalent coming from places where people, need to have a certain number of papers in order to get tenure. And so they hire a company. You can pay a company to generate papers for you without you having to actually, generate data. And of course, this is not the way science works, but it is becoming a tremendous pollution problem in the scientific literature. So what they did was they focused specifically on papers that have come out in PLOS One, not because PLOS One is uniquely bad, but because PLOS One is very open about allowing access with an application program interface and API where people can mine the data from it, which made this type of analysis possible. And it's also, kind of the 800-pound gorilla in publishing, lots of stuff coming through there. And they worked out this whole, analysis, and you can read the science article, and you can also read the paper on it, and it's pretty damning, I think.
Paper mills.
Yeah.
Wow.
And a big problem with it is the industry itself is not illegal.
Right.
It's very hard to find laws that are being broken here because they're not misspending grant money because there's no science actually being done. The companies are providing a service, which is not illegal. The authors are, presenting these papers to the journals. The editors are putting them in, the editors, these networks of shady editors are getting them into the journals. And kind of hard to pin them with some kind of charges, but this is obviously a problem.
So I guess lying isn't a crime, depending.
No,
on the
lying
situation.
is not a crime in and of itself, which is a good thing. You can, gee, Aunt Sadie, this pie tastes great. That
Yes,
doesn't
that's
get you
sort of lie.
arrested.
Yes.
But this is a little beyond that, and hopefully something gets done about it.
It's interesting. Yeah, you submit a study that was never done. I don't know. Even if there were a statement in the journal which said everything has to be stuff you actually did, it wouldn't matter. There's no law enforcement.
Right.
I mean, worst that would happen is that, they could get their paper retracted.
Which apparently happens a lot with some of
And
these
that's actually what they tracked here was the percentage of papers being retracted, and linked that to, certain editors had a real high rate of retraction.
That seems like the only thing that can really happen. And depending on who you are as a scientist and where you are, that may or may not be a disincentive for you.
So when I first saw this, and you said, Alan, that, there's pressure to get tenure and grants. When I was in academia, well, I still am, but I don't do research anymore. I went through this, right? I've never thought of cheating. I always thought if I cannot do it, if I cannot achieve tenure, if I can't get a grant, then I'm not suited for this and I have to do something else, right? I would never have cheated to achieve that because it sticks with you the rest of your life.
Yeah.
That wherever you got, you cheated. That's horrible. Don't you think?
Yeah.
Yeah.
But some people don't care. I cheated. Now I'm tenured. Thank goodness. And then they forget it. Oh my gosh. So.
And depending on, what's been normalized in people around you, you may get the idea that this is what everybody's doing when it's not.
When I was a professor, a very young professor, and I heard incidences of fraud, it was always stunning to me that people, because I had this, pearly view of what was going on around me, that everything was fine, and then there's fraud, and it's like, why would somebody do that? I would never make up data. If you can't do it, get out.
If you don't love it and sort of have the integrity of wanting it to be right.
Yeah.
Then you're in the wrong business.
Yeah.
Yeah.
Exactly right. But a lot of people don't feel that way. It's called integrity. Yeah.
Mm-hmm.
All right. 2 other news items. Here is an article in CIDRAP. Report suggests FDA may not reauthorize Pfizer COVID vaccine for young kids. So right now, the Pfizer COVID vaccine is under emergency use authorization for kids 6 months to 4 years old, right? And the FDA is saying, we might not renew that. And you know who's back at the FDA in the vaccines division? Vinay Prasad, who just left 2 weeks ago. I have my pick is all about him. So this would be crappy because the Moderna is only approved for kids with 1 or more health conditions, and Novavax 12 years and older. And in this 6 months to 12 years of age, there's a lot of pediatric deaths. I mean, it doesn't, even if there were 1, it would be too many, but there are many, and this doesn't seem right. I don't know what the problem is. Here's some numbers. Well, anyway, that's Pfizer and Moderna. We have some numbers. Yeah, the uptake of vaccines in this age group is low. And these kids who die are typically not vaccinated. That's what Daniel is always talking about.
Well, and part of the problem here is that the Pfizer vaccine is actually the only one that's available for kids from 6 months to 4 years who don't have other health conditions.
Yep.
And so if this vaccine is not renewed, then there will be no vaccine. It's not as if the Moderna vaccine or some other vaccine would still be an option. this is the only option right now for this group of kids.
And I mean, the idea that the fact that the others, say Moderna, is only approved for kids in this age group if they have some complicating condition, suggests to me that there's been a little failure of logic here.
Yes.
Because if it's okay for a sick kid, why is it not okay for a healthy kid to get the Moderna vaccine?
Right.
That seems like something that could be fixed, but likely not under this administration.
Makes a lot of sense. Yep. But we don't do logic in this administration.
No.
When you lie, it's hard to have logic.
Yeah.
Right. And the final is the New York Times article, which is paywalled. So I have to look on my laptop here. Draft of White House report suggests Kennedy won't push strict pesticide regulations. So this is a White House report on the health of American children. Will stop short of proposing direct restrictions on ultra-processed foods and pesticides. And RFK Jr. has already said these are a major threat to health. Right through his Make America Healthy baloney again. That's not baloney, but that
is in fact the thing that people who seem to really like him tend to like him for most, if it's not anti-vax stuff.
Yeah. So the White House is not going to regulate these because it's a money issue.
There's a lot of money on the table, and a lot of the companies that make the ultra-processed foods and the pesticides and the agribusiness are big Republican donors.
Yep. It's you go. Money controls it all. Unfortunately. Anyway, I'm not going to read any more of that because that's the gist of it. Of course, it is a draft and it could change. All they have to do is put it through ChatGPT and it'll change. But they're not because it's all about money. Okay, we move on now at 17 minutes. Not bad.
No.
We move on to our science. We have 2 papers for you. The first, we're going back to visit T cells and COVID again. Glad you're here, Brienne, because it would be like Alan and I discussing
fumbling
T-cells,
around with immunology, wishing Brienne was here
without a license. It's a Cell article. Highly conserved betacoronavirus sequences are broadly recognized by human T cells. This is from the La Jolla Institute for Immunology in La Jolla, Murdoch University in Perth, Australia, the Ventnor Institute in La Jolla, NIH in Bethesda, UCSD in La Jolla. And we have, let me see, this first name is lovely.
Yes.
Tertuliano Alves Pereira Neto. I wonder what is that? Do you think it's Brazilian?
I think maybe. It looks
NATO
Portuguese to me.
Yeah, but NATO seems Portuguese. Anyways, Zmasek, Avalos, Sydney, Trevizzani, Phillips, Malal, Fraser, Tan, Showerman, Sette, Alessandro Sette, who's been on TWiV twice, and Alba Grifoni. So, this is about pandemic preparedness, which apparently the administration is not interested in because it canceled a lot of NIH grants about pandemic preparedness, but we should still focus on it. And so the idea is that you say, we don't know what the next pandemic is going to be. No one knows, but maybe we could have a focus on a family of viruses. This family just brings up RFK Jr. He said, we're making vaccines to protect against the whole phylum.
Yes.
Mm-hmm.
The whole phylum. Do you know how high phylum is on the taxonomy?
When I read that, I laughed so hard. And I also was like, well, if you had asked me before this, I would not have thought that he knew the word phylum.
Yeah.
So phylum would be like all the coronaviruses and more, right? So that would be all the coronaviruses and all the influenza viruses. There are multiple phyla. I actually looked it up. It's on the other computer. You go to ICTV and you could look up how high phyla is. I think there are 22 different phyla.
Only kingdom or domain are higher.
Yep.
Yeah, it's kingdom and domain. Realms are higher too, right?
It's realm instead of domain, right?
Yeah, I'd like to be the master of my realm of viruses, wouldn't you? Realm is a great word.
Yeah.
Anyway, we're going to look at broadly protective approaches to pandemics. We don't know what's going to emerge, so we have to make broadly protective vaccines. That would be the idea, right? And so here we're talking about betacoronaviruses, including SARS-CoV, MERS, and SARS-CoV-2, and also the common cold coronas. And then within this, you have subgenera Mbecovirus, Sarbecovirus, Merbecovirus, and Nobecovirus. It sounds like Soho and NoHo
Yeah.
and whatever we have here in New York. They've been linked to previous outbreaks, so they're potential zoonotic pathogens as well. So, for SARS-CoV-2, people are exploring broadly protective approaches, like the antibodies against the receptor binding domain of the CoV-2 spike can induce broadly protective antibodies, and so can other parts of the spike protein. But here in this paper, we're going to focus on T cells, not antibodies. T cells do limit disease severity. We know that. We've been talking about it for 5 years, right? Mm-hmm. Early T cell responses are correlated with favorable clinical outcomes, reduced viral load, and even in the absence of antibodies, T cell can protect against severe disease. We've done those papers here on TWiV. And, neutralizing antibodies may decrease over 6 months, but T cells stay high. And so they're critical. And even in this one challenge study, protection has been shown to be mediated by T cells in the absence of neutralizing antibodies. But the cool thing also about T cells is that they can be cross-reactive because the epitopes are conserved compared to antibody B-cell epitopes. We already have evidence that infection with other coronaviruses may provide some protection against SARS-CoV-2 via T cells, right? Even common cold coronaviruses reduce disease severity and so forth. So here they're going to say, can we develop a modality where we find conserved, we identified T cell epitopes, the peptides that would be T cell specific, be recognized by T cells. Can we find common ones across the beta coronaviruses and show that they are actually cross-reactive?
One of the other reasons why T cells can be really important in this sort of situation is that, and specifically in having broad responsiveness, is that we can have those cross-reactive epitopes, but also because T cells can respond to epitopes that are internal to a microbe, particularly internal to a virus. They're not quite as focused on, say, something like the spike protein that is always on the outside of the particle. Mm-hmm. And as a result, those might be parts of the virus that are under more structural constraint, and that are harder to change than some of the external parts that may be a little more malleable.
And that's because T cells are going around to cells saying, papers, please, and looking at what's going on inside that cell through their receptors.
It's like an airplane, right, Alan?
Yes, right.
Parts can be shot up. It's okay.
Yeah. So we're going to be talking a lot about these epitopes that will generate a T cell response that are conserved epitopes, and they call them conserved T cell epitope regions, or CETRs.
Yeah, but you almost said episode, didn't you?
Episode, yes, right.
This is epitope 1245.
Epitope 1245. So we're going to be talking about CETRs, and that's these potential conserved regions of beta coronaviruses that would react to T cells, or the T cells would react to.
Conserved, what is it?
T cell epitope regions, CETRs.
T cell epitope regions, right. Okay, so what they do first is to look for regions that are CD4 and CD8 T cell epitopes. There's an epitope database. Did you know there was an epitope? It's great. There's an everything database.
I did know about this database.
Now, they're looking at first at SARS-CoV-2, right? And they find that the S protein has highest number of identified epitopes, probably because it's been studied the most. 414 CD4 and 386 CD8. Nucleocapsid is close behind. It's a non-structural protein, right? On the interior of the particle, 172 and 141 epitopes. Then another viral protein, NSP3. And they go through all the viral proteins, and they end up with a lot of epitopes, almost as many as TWiV epitopes, I think, in total.
Yeah, I don't remember off the top of my head the sizes of all of the SARS-CoV-2 proteins, but you would expect that there would be also a lot of epitopes in S because S is really big.
Yeah.
And so I don't know if this, correlates exactly with size of protein and number of epitopes, but it seems in the right ballpark.
The other thing that's important is that these epitopes are class 1 or class 2 restricted, MHC class 1 or class 2. And, class 1 and class 2 genes are polymorphic in human populations. And so you don't want to just have epitopes that are found in New York.
Well,
right?
Actually, that would be a pretty good sampling of humanity.
New York would be.
You don't want epitopes that are only found in like one isolated branch of the population that's got a particular.
Yeah. So they find among these epitopes, they are found in. They're restricted to 68 class 1 and 85 class 2 alleles that are balanced among the U.S., Europe, and China. So that's good. And we're going to get back to that again. We're going to talk a little bit more about that. All right, so now we've identified these T-cell epitopes on SARS-CoV-2 proteins. What about other beta coronaviruses? So they look to see in the genomes, the proteomes of other beta coronaviruses, and they can find conserved epitopes. Then they look at the immunogenicity potential. So there's a tool, the Immunome browser tool, that you can use to look at immunogenicity of peptides. And so they put those two together, the conservation and the immunogenicity, and they end up with the peptides that they're going to study for the rest of the paper. So the S protein had 4 CETRs in the S protein. So they make overlapping 15 mer peptides corresponding to each CETR. And they also have non-CETR, non-conserved regions as controls. And these are all done using the ancestral coronavirus. And then they look at how these activate T cells. Right? They look at markers of T cell activation-induced markers, AIM assays, which we've talked about before. And they have T cells from 29 adults, right? Which it's not going to be hugely broad in terms of MHC, but I guess you'd have to have a lot.
They do show they have a table on the demographics, so it's not like 28 white guys. It's a mix of male and female, 58% white, 17% Hispanic, 17% Asian. So it's a little bit of diversity.
Yeah, a little bit.
And I was impressed, and this makes me almost feel old, by the fact that, I looked at their computational data and I was like, gee, a database of immunogenicity. Like, don't you have to do some experiments? I don't know. And I liked that the 4 CETRs that, really seemed to be the ones that had high levels of immunogenicity were in fact immunogenic in real experiments.
Yep. So they ask whether the T cell responses against these spike versus non-spike CETRs had increased cross-reactivity with other coronaviruses. So they pick a virus from each subgenus, right? OC43, HKU1, HKU9, MERS, more and more. And for each virus, they make the corresponding peptides from the region that is conserved in the spike, right? They make pools of them, and then they test whether T cells respond to them from these patients by this activation-induced assay that we just talked about briefly. And so essentially, these spike CETRs elicit T cell responses that recognize multiple viruses, suggesting that there are conserved epitopes among these beta coronaviruses, spike in particular, that promotes T cell cross-reactivity in people so these people have been exposed to SARS-CoV-2 or they've been vaccinated before. And that's good because we're looking at spikes so far. So beyond the spike, they look at other proteins, of course. They look at the N protein, which has 2 CEDARs. They have non-structurals, so like NSP3 and others. They see CEDARs in all of these proteins. So basically, they end up with 1,326 CEDAR residues. 20% of the considered proteins have these conserved epitopes. And so next they ask whether these there is cross-reactivity at the single epitope level. Before we were using pools of peptides, right? So they make they make T cell lines using the peripheral monocytes from these donors who are known to respond to specific SARS-CoV-2 epitopes in different proteins and in ORF7A, and then they add individual peptides to these cultures and see if they respond by making interferon gamma, ELISpot assay, right? And so, yes, they find cross-reactivity for 6 sarbecovirus homologs for these proteins. And so, but not all of them that they identified before. And they say this shows you that on a single epitope, the conservation can vary considerably functionally. So, and that's why it's good to have a lot more epitopes in the well, here when you're assaying for them. But eventually, if you're going to immunize people, you want to have a broader immunization than just 1 or 2. Next, they looked at whether these these CEDARs, how do these CEDARs compare to the full-length spike used in the vaccine platform? And so they again took the lymphocytes, whether from these patients, and they found that the non-spike CEDAR is less immunogenic than the spike CEDARs, interestingly. And then they asked whether these non-spike epitopes are able to further increase the T cell cross-reactivity as compared to T cells just recognizing the spike. So we know the spike has some cross-reactivity. Does it get better if you add non-spike epitopes? And the answer is yes. T cell cross-reactivity increases and you get broader immunity against diverse species when you add non-spike epitopes.
I thought it was interesting that they seem to really get that improvement in CD4s here.
Yeah.
Whereas the CD8s seem to be really critical previously.
Why do you think that's interesting?
I think that, well, one reason why it's interesting is that there are fewer different alleles of MHC class 2. And so if you can get diversity in MHC class 2 and turning on CD4s, you're going to impact a larger proportion of the population. But I also just think that our general immunological knowledge of sort of reactivity of CD8s versus CD4s and triggering stimuli and you know, things like that are still things that we have questions about.
So then they get back to this population polymorphism of HLAs. You know, you want to avoid ethnic bias, right? And have as many as you can. Have the epitopes work in as many populations as possible. So in other words, the coverage of HLA polymorphisms by your epitopes is important. So they predicted the population coverage provided by each epitope sequence. And they have a way to do this. Obviously, it's a prediction. It's an algorithm. It's not an experiment. And they look at the population coverage with full S versus these non-S CEDARs, right? And if you put the CEDARs non-S with the S epitopes, you get more than twofold higher population coverage for both class 2 and class 1 when you add the non-spike. So that's another reason why, not just for cross-reactivity, but you're going to get a better population coverage when you put the non-spike epitopes in. They also did this, so that was a computational study. They also did it with their cohort of 29 people, right? And they could type 27 of them for HLA. And basically, again, they get a twofold higher, what's the word I'm looking for? You get a twofold higher coverage of HLA types when you include the non-spike epitopes.
Right.
This I was really impressed by this because the HLA coverage is always the problem, and HLA diversity is always such a problem with T cell biology. And so really being able to hit this many more HLA types isn't just great for ethnic diversity, it's also great for like diversity within a group because there's so much HLA diversity.
Yeah.
Yes. So between the three of us, do we all have different HLAs?
Yes.
Yeah.
So it's almost on a person by person basis.
Yeah, especially, particularly if you get to what combination of HLAs people have, but some of the the HLA-Bs, for example, I don't think that we've gotten to 1,000 alleles, but I think like 800 might not be out of the question. And so to think that you and I and Alan each have 2 of those 800. And so we certainly, even if we share one, we probably have different pairings.
Yeah.
And then multiply that by 2 copies of HLA-A and 2 copies of HLA-C, both of which are quite diverse. And all of the DRs, DPs, and DQs, which are not quite as diverse as class 1s.
So you roll an 800-side die twice.
Yeah.
And see if anybody gets the same 2 numbers, and then that's just for one locus. Yeah.
Yeah, and so the fact that they increase how many people win with that 800-side dice here by this much is pretty cool.
Yeah.
Now, despite this, these nice results, some seeders have low representation in the population, and so they decide to take these peptides out. So the mix is not as complicated anymore, and you can also so you can optimize it, the number of different epitopes you have in this mixture.
And ultimately, what they want to get at is a formulation you could put in some kind of a vaccine that would be a certain number of antigens you'd give people that would hit these seeders, stimulate good responses. And the problem there is if you put in a whole bunch of them, you may have something that's immunodominant that all the T cells glom onto that distracts them from paying attention to the others that would have been good ideas.
Well, or the other problem is that you have to think about what the dose is. And so if you use, 10 peptides and you give a gram, then you're giving 1/10 of a gram of each. And if you give 100 peptides and you give a gram, then you're giving 100th of a gram of each. And so by limiting the number of epitopes, then you can actually make sure you're maximizing the dose of each epitope.
Yeah.
Yeah, we're going to talk more about that later in terms of making a vaccine. Anyway, they remove these low coverage region. It doesn't really impact the population coverage in terms of HLA types. It doesn't impact the immunogenicity either. So they say we should focus on high coverage regions outside of spike in addition to spike. And this gives us good population coverage, and it maintains immunogenicity just as good as using the whole collection of epitopes with high and low coverage regions. So you don't need the low coverage regions. That's good. So that's the work. It's a framework for future development of vaccines. So you put these non-spike seeders, T cell epitopes with spike. You get good population coverage. You get good immunogenicity and cross-reactivity of T cell responses. And so you could use that data to make a vaccine. So what would the vaccine look like? I mean, is this a peptide mixture basically? Or so because if you put the whole proteins in, first of all, there are too many proteins, and you're going to have regions that are not needed, right? In terms of both what Brienne said in terms of dose and also in terms of immunodominance. So is a peptide framework the way to go here?
Maybe a peptide framework. When people have thought about vectors or nucleic acid vaccines, there's also been some idea of a mosaic where you basically are making a piece of nucleic acid that just encodes for each of the peptides in a string. And those peptides will get degraded differentially to become epitopes in different people.
Yeah.
And so I think that the mosaic approach might be one way of thinking about this, maybe making a mosaic mRNA vaccine, making a mosaic adenovirus or other viral vector or something like that.
So in this paper, do they use the whole spike? Because they're talking about full spike. Or are they talking about all the spike epitopes as peptides?
I think they were talking about full-length spike.
So that would give you the neutralizing antibodies as well, right?
Right. And that is the problem with the mosaic is you're just giving the T cell epitopes and you're not going to actually get this Franken protein to fold into anything.
So you probably want to have some neutralizing antibodies as well, right? Even though in some people, the T cells may be enough, you can't assume it's going to be good for everyone, right?
Right. I also just want to make a correction.
Yep.
I'm sorry, Jonathan Udell. After I said it, I decided I would Google numbers of HLA alleles. With HLA-B, I said I didn't think there were 1,000, but 800 sounded legitimate. There's 7,561. So I was a little low.
Off by almost an order of magnitude. Okay.
Yeah.
But in the right direction, because the point you were making is there's a lot.
Exactly.
Yeah.
In fact, I don't know if anyone noticed it. I'm pretty sure I made a face when this Google came up.
So there you go. You've got cross-reactive T cell epitopes that are going to be working in most of the population in terms of HLA types, and put that in a vaccine with full-length spikes somehow. So they have a nice discussion here, which actually Brienne already touched on, but we'll go over again for those of you who weren't listening. Maybe you went off to make tea or something. So there's a correlation that they see between these conserved epitopes and immunogenicity, right? And so they say that these highly conserved regions are preferentially recognized by T cells, probably because of evolutionary constraints. These are structured regions that can't change very much, N protein and some of these non-structural proteins, whereas spike has regions that can change all the time, and the structure is maintained. So they say this evolutionary stability ensures that immune recognition of these epitopes remains consistent across diverse viral variants. And that was our theme throughout the pandemic, that as spike changed to evade antibodies, the T cell epitopes were mainly conserved.
Yeah.
And it saved probably many lives.
Yes. And I remember when I first heard about the vaccines being spike only, I was very worried because of that. I wondered, will this be enough T cell epitopes? And then I found out what a big protein spike was, and I was like, oh, right, that's fine.
So we were lucky that it was so big.
Yeah.
Remember we had an episode called Don't Put All Your Spikes in the Same Epitope Basket?
Or maybe eggs in the spike basket or something.
Something like
Don't
that.
put all your eggs in the spike basket, because we were skeptical. But it worked.
It worked.
Yeah.
Yes, it's a very good vaccine, despite naysayers who say mRNA vaccines are not good. They are. It worked. They're very protective.
Yeah.
Saved millions of lives.
I do have one tiny little nit to pick with this paper if the authors are listening. There is a repeated typo where they use homologs instead of homologous. So they're homologs proteins and homologs regions, and it just kept coming up.
Oh, well.
Anyway.
Next paper, Cell Reports, identification and engineering of potent bispecific antibodies that protect against herpes simplex virus recurrent disease, from Gilead Sciences. Isn't Gilead the company that makes lenacapavir?
Yep.
Yeah, we mentioned Gilead previously.
We were kind of ragging on Gilead.
Yeah, we were picking
Yeah.
on Gilead. Now we're going to do a paper from them at University of Cincinnati.
Yeah, we got Lee, Viadu, Clemakar, Bernstein, Pei, Yu, Wang, Bravo, Ding, Sito, Hung, Yu, Xing, Papalia, Khan, Carr, Thomas, Tong, Desai, Jarus, Mercier, Holdorf, Simon Fletcher, and Emma Abernathy. Herpes simplex viruses. Types 1 and types 2 causative agents for oral and genital herpes. High prevalence globally. So HSV1, 67% of people infected, 13% for HSV2 under 50 years old. And I didn't know this one. The economic burden of genital herpes alone is $35 billion. That's 2016. So you're saying, what is the problem? Well, it's painful, and you get psychosocial distress, right, with genital herpes. So that's very expensive. You also have severe disease associated with infection like in immunocompromised people, encephalitis, keratitis, and neonatal herpes. These are horrible infections. And HSV2, having HSV2 triples your risk of getting HIV infection. This is a good one. This is a paper we got to do in a couple of weeks. HSV infection doubles the risk of dementia in older adults. Wow. How about that?
Yeah, I mean, doesn't it sort of seem like at this point it's a lot of things in the herpes virus group between, your shingles vaccine and dementia and things like that?
Yep.
Anything that gets in your brain is probably going to...
Probably not something you wanted there.
Yeah. I mean, yeah, anything that's getting in your brain and doing things is probably going to contribute to dementia. And I think by thinking a lot, you lessen your chance of dementia.
Maybe.
That's good. Anyway, there's no vaccine. But we are limited to therapeutic options like nucleoside analogs, acyclovir, valacyclovir, famciclovir. But, you can use those if you get a cold sore, and it will accelerate it from going away. It will accelerate its disappearance, but they don't prevent recurrence because the DNA is in your neurons, and periodically it will reactivate and make virus and give you cold sores or genital herpes. So we do need therapeutic options. So by the way, you get infected at a very young age, close to birth or shortly thereafter from your parents' saliva. You get infected, the virus goes latent in your ganglia and then periodically reactivates. So you get cold sores or genital herpes. You're stuck with it for life.
Cold sores are the ones, the oral infection is the most likely you get from your parents.
Yes.
Yeah, the
genital infection you get
you're not getting
typically
those from
later
your
in
parents.
life.
Thank you for listening, Alan.
Yes, I
but-
just wanted to point that out.
And the cold sores could also come from, sharing cups and other kind of mucosal secretion contamination if you don't acquire it early in life and from parents.
So this is Saul Silverstein taught me years ago, unlike love, herpes is forever.
Yes.
He was so cynical, wasn't he, Alan?
Oh, yeah, I loved it. It was great.
He was just a loud, cursing, cynical man. Well, he still is. He exists. Okay, so now this paper, we're going to try and make a therapeutic. And we're going to use antibodies, and we need a little background on the viral proteins that we're going to target. So the viral genome encodes 4 glycoproteins that you need for virus entry. Very selfish virus needs to have 4. SARS-CoV-2 just needs 1. So we have the receptor binding protein, which is called GD. We have 2 regulators of fusion, GH, GL, and then we have the actual fusion protein, GB. Now, the other complication here is that during infection, the virus moves from cell to cell between tight junctions. And so it can evade antibodies that way. So that's called cell-to-cell spread, CCS. And we know that GB and GD are important for this cell-to-cell spread. And we also know that GD engagement of its receptor is important. And we can make antibodies that block cell-to-cell spread, but these are very rare antibodies in animal models. Further complication is that the virus has an antibody evasion mechanism. They basically make 2 other glycoproteins, GE and GI, which bind immunoglobulins and internalize them and degrade them. So that's another way of problematic way to get around this.
And during this cell-to-cell spread, I mean, Vincent said they're going through tight junctions, which, as the name implies, this is an area where the cells are very close together. And the thinking is that it's hard to get antibodies in there to catch the virus between cells And so that's another
Yeah.
issue is
Because it's
just
a small space.
yeah, our antibodies are too big to get in there, which may be one reason with this happens so rarely.
Mm. So we know that blocking this evasion mechanism can help vaccine efficacy when you're in trials or testing vaccines. So they think maybe this is an important part of a therapeutic antibody. So let's find some antibodies that can block cell-to-cell spread and also evade this antibody targeting, or antibody-targeted degradation. So let's see if we can do that. Now, in terms of other neutralizing antibodies, 3 have been evaluated in human clinical trials. They're safe, they're well tolerated, highly efficacious in mouse models, but one of them failed to demonstrate superiority over standard of care in phase 2 for recurrent genital herpes. All 3 are antibodies that can strongly neutralize virus, but they don't block CCS as well. And so the authors say maybe that's what's lacking here. Maybe we need to have a good antibody that blocks CCS. And so here comes camelid variable heavy chain region antibodies, or nanobodies. They're small antibodies that can bind difficult to access epitopes because they're smaller.
And we've talked about nanobodies on TWiV before.
We have. I like to talk about camelids because just to say alpaca is a lot.
Yes. Alpaca and llama are cool.
Very cool animals. And if you've ever seen some, they're very interesting. So the tight junction issue has been mentioned already, may be a challenge. So maybe these nanobodies can get in there, right? So that's what someone had an idea and they said, let's try it. And that's what we have in this paper. Let's make small antibodies. So they immunize an alpaca, or maybe more than one. I don't know. Probably just one. One alpaca is large. You can get a lot of antibodies. And then they discover antibodies that not just both neutralize and block CCS and target the right glycoproteins. They end up with 43 GB binding and 34 GD binding antibodies, and they get the sequence and they fuse it to human IgG-Fc, and then they do neutralization activity. All but 2 of these bound very well to the target, and 14 anti-GD, 6 anti-GB. These variable heavy FC antibodies neutralize lab strains of HSV1 and 2 pretty well. They also looked for inhibition of cell-to-cell spread, and again, they find good numbers of anti-GD and anti-GB antibodies that inhibit cell-to-cell spread. And so now we're going to be studying one of each of these representative nanobodies in this paper, and they're going to be comparing them to what they call tool antibodies, right? Tool against GB or GD, tool GB and tool GD, which have previously been described, but are not great at blocking cell-to-cell
Right,
spread.
and they call them tool antibodies because they're commonly used as tools in herpesvirus labs, right?
Yeah.
I don't really like the name. But
I did not like that either.
It's weird.
No, it sounds like the antibody's picking up a saw.
Well, to me, it sounds like if you call someone a tool, that
Right.
it's derogatory, and these antibodies should not be called derogatory.
No, well, I also it took me a little while to figure out that they weren't like modified in some way as some like interesting antibody tool, not just an antibody.
Yeah.
All right, so they, so as I've said, they picked the 3 most potent nanobodies targeting GB and GD, and they look at, they pick them because of high neutralization and high ability to block cell-to-cell spread. And then we move on. Then they said they make bispecific antibodies for 3 of the most potent CCS blocking antibodies. So bispecific means they're going to have 2 different specificities in the antigen combining region.
Yes.
And they have.
So usually an antibody is symmetric. Has the 2 arms of the Y, and they are identical and bind the same thing twice. And so here, what's really cool about these and all other bispecific antibodies is that for the purpose of a therapy, not something that you're going to have made in an organism, you can have an antibody that is not symmetric, where the two arms on either side will bind to two different things. And that can be cool for a lot of reasons. There are a bunch of these that are being used pharmacologically.
So the nanobody is just one chain, but they have put a human FC to make it bispecific.
No,
right?
the nanobody is actually has a heavy and has a variable and a constant region.
Mm-hmm.
It doesn't have a light chain.
Okay.
So they've taken the variable region from the nanobody,
variable heavy, and put an FC.
and then they've put on the human
Okay.
rest of the human heavy chain. So
Got
it's basically it doesn't need a light chain. So it's the human heavy chain, alpaca variable region, no light chain needed, because
So
the alpaca
it's-
one doesn't need it.
So it's still smaller than a conventional.
Yeah, because it
Yeah.
doesn't have a light chain.
Yeah. But aren't there nanobodies that are single chain also?
There are single chain that are just the variable region,
Yeah.
which is nice. I think the reason why they go with this, because at first I was like, why do you go with this instead of using just the variable region? And I think that they can probably get better blockade by having a little bit of the bulk of the FC portion. They can get some more FC mediated function.
Yeah.
But they have to use the human FC portion, not the alpaca FC portion, because they don't want to introduce that foreign protein and potentially have people making a response to that foreign alpaca FC.
Right. And of course, this allows you to make bispecific. They target two different proteins, viral proteins as well. You couldn't do that on a single chain. So they make bispecific for 3 of the most potent antibodies using 2 different valency formats. So they have a monovalent heterodimer, 1+1, with 1 binding site each for GB and GD. And the second is a bivalent homodimer with 2 binding sites each for GB and GD. There's a figure here that shows that. And so the 2x2, the 2+2 format, the bivalent, was twice as potent at blocking cell-to-cell spread compared to the monovalent heterodimer.
And by the way, both these papers today are open access.
And several of these had an improvement in CCS blocking over their most potent monospecific derivatives, right? So being bispecific is really good. It really helps. So, for example, the most potent bispecific antibody had a CSS blocking potency improvement over the tool antibodies of 23 and 34-fold, respectively. So there's an increase in potency when you use 2 different antibodies linked in a bispecific format, at least for the epitopes that they've looked at, right? So then they say, well, does the orientation of the binding arms in this 2+2 format affect potency? So they swap the order of the anti-GB or the GD arms, or tether the anti-GD to the FC region, and they look at the cell spread potency, blocking potency of these. The order doesn't matter. Doesn't matter at all. They tested if the same potency enhancement is seen when 2 different VHHs are physically linked in a 1+1, and 2 of these are unable to block CCS. So you need to have bivalency, but they still neutralize. So cell-to-cell spread is a unique process to block here. Then they do a series of experiments to try and understand why is a bispecific format better than doing monospecific. They find that, in fact, you need both GB and GD to be targeted to achieve the potency boost, and you have to have both neutralization and cell-to-cell spread blocking ability. So even if you just had cell-to-cell spread, you can't have it without neutralization activity.
Yeah, and the key thing is that if they use the monospecifics, then one antibody might bind to one viral particle, and the other antibody might bind to the other viral particle. So you have one viral particle where GD is really well blocked, one where GB is really well blocked, but in each case, the other protein is available.
Right.
You can't make sure that those two antibodies you deliver are both going to bind to the same viral particle, I guess, unless you gave a massive dose. But with the bispecific, this antibody is now going to block GB and GD on the same particle. And in fact, the GB and GDs that are right next to each other on that particle
that's right.
to give you this blockade.
Right. So we need to have both glycoproteins in these antibodies to get CCS blocking. So then they wanted to see what this looks like, because it's always nice to see things when you can. And so they solved the structures of these antibodies in complex with GB or GD. So they have the most potent bispecific antibodies, and they do cryo-EM and see how they're binding to GB or GD. Now remember, what they're looking now at is the post-fusion state of these glycoproteins, the ones that are catalyzing fusion. Remember, there's a pre-fusion and a post-fusion state. And so they looked at the post-fusion state of their GB trimer binding to these antibodies. And they find that the D and the two different antibodies that they're looking at bind in epitopes that are opposite ends of the post-fusion structure. But when they do modeling of the pre-fusion structure, right, which is presumably when these antibodies are going to bind. So on the virus particle floating around in fluids, the glycoproteins are in a pre-fusion state because they haven't bound a receptor yet and haven't fused yet with the membrane. So the pre-fusion state, and that's when the antibodies are going to hit them. And so they find that in the pre-fusion state, likely these epitopes are pretty close. And so that's how this bispecific antibody can hit both epitopes at the same time. Did I get that right?
I think so.
Yes.
They say potentially explaining how disparate regions in the post-fusion structure can be targeted by both bispecific antibodies. So they have two different bispecific antibodies. D76 and D71, which are not really telling you anything. And that's why I have to always figure it out de novo. Then they find that these epitopes, so D7, one of these bispecific antibodies, and also the tool anti-GD have overlapping epitopes with each other, and they also overlap with the binding surfaces described for entry receptors, Nectin-1 and HVEM. Nectin-1, it's the poliovirus receptor.
Yes.
It's so funny. Many years after we found it, Pat Spear found it was also a receptor for herpes viruses. So anyway, these antibodies, where they bind, partially blocks the receptor binding, and that's probably an important part of how they block infectivity and maybe also cell-to-cell spread. And by the way, these epitopes that we're talking about here, the GB epitopes here, and the GD epitope are conserved 99% conservation across HSV1 and 2, 945 sequences they looked at. So that's good,
Yes.
right? Because it suggests that it will be effective against many different isolates. So they ask how broad spectrum are these antibodies? So they have 4 lab strains, 2 HSV1, 2 HSV2. They have 8 clinical isolates, 4 of each, and 3 of those are from patients with acyclovir-resistant viruses. And so they do neutralization assays with these bispecific antibodies. All the clinical isolates were neutralized just as well as the lab strain, so they clearly have broad-spectrum anti-herpes virus coverage. But that's neutralization, right? What about CSS? Am I doing that next or not? No. Oh, okay. But so they say all this work that we've done has shown us where the epitopes are, but they still don't explain why bispecific is better. And so they say, because the epitopes are close, maybe the antibodies bind simultaneously, thereby and cross-link the entry process. And so they use Biacor, a way of measuring affinity, to look at this, and they find that the antibodies bind sequentially to GD and DB. So they bind both glycoproteins at the same time. So their model is the bispecific antibodies bind GB and GD, cross-link the two glycoproteins. That blocks fusion because it blocks receptor binding, which in turn blocks fusion. All right, so, and that's in line with the predicted location of these epitopes in the prefusion state. All right, so that's one part of the problem here. You make a nice bispecific antibody that hits both glycoproteins, and you can neutralize and prevent cell-to-cell spread. But then there's this other herpes virus immune evasion mechanism where the viral glycoproteins bind to the FC portion of the antibodies, and they get internalized and degraded. It's kind of like binding to an FC receptor, right? Mm-hmm. So they said, why don't we disrupt the FC? Why don't we change the FC portion of these antibodies to disrupt their ability to be bound by these viral glycoproteins? And so it's already known what amino acids can be introduced into the FC region to change their half-life. They're called the YTE change. And so they put these into their idea is, okay, these changes in the FC could potentially abrogate the binding of the FC to the viral glycoprotein. So they introduced these changes into their bispecific, into their tool antibodies first. And they find that that introduction of those changes causes a 700-fold lower binding affinity to the viral glycoprotein GEGI that are involved in the immune evasion. And they also put them into their bispecifics, and it also reduces their binding to the viral glycoprotein. And they do this in cells as well. So this is a cool approach, right? So if you want to evade evasion, you change the FC part of these antibodies. And so if you're going to use these therapeutically, you would give people the modified versions. Right. And the viral glycoprotein wouldn't bind to it and wouldn't bring it in cells to degrade them.
Yeah, so the virus has these proteins that will grab the antibody, bring it into the cell, and degrade it. And here they're removing the handle from the antibody, so it can't.
Well, they're changing the shape of the handle.
Changing the shape of the handle, yes.
Because we still have an FC portion. It's just the FC portion can't be bound by the viral glycoproteins.
Right.
And so all of this is sort of coming up with this highly engineered antibody-like protein or nanobody or whatever you want thing.
Thing 1 and 2.
We're only able to do. Yeah, it's thing 1 and thing 2 bound together because it's bispecific.
Yes.
And it's only possible because of how much we know about the biochemistry of the antibody protein that we can make all these cool modifications and make this highly modified antibody thing. And so again, yay for basic research and the things we learn. And also yay for looking at a non-model organism, the alpaca and the llama, to find out about these uniquely structured antibodies.
Yes.
But Brianne, we're going to give infectious diseases a rest for 8 years, you know? We don't need to do any of this research anymore. See how stupid that statement is? This is such beautiful stuff here.
That's why we should do the basic science. We're not trying to cure a disease. We're going to learn this basic science, but look how much it's going to give us.
Yeah, we'll fool them. Yeah. We're not doing infectious disease research. We're doing basic science.
Because they love basic science.
Oh, yeah. That's not going to get you any more traction. I'm sorry.
But as long as you don't have keywords, because that's all they can do is search for keywords in your grant. Okay, so then they actually show that these changes in the FC portion do reduce internalization of the antibodies into the cell. So that's all good. Now, what else does the FC portion of an antibody do? It mediates effector functions like binding to cells like NK cells, right? So they ask whether some people have found that antibodies that are involved in effector functions like antibody-dependent cellular cytotoxicity are important for controlling herpes simplex infections. And so they look at this, they ask whether these FC changes that they've introduced into their bispecific antibodies, whether they have any impact on antibody-dependent cellular cytotoxicity. This is when an antibody is bound to an FC receptor on like an NK cell, and then the NK cells will kill whatever the antibody is bound to, right?
Mm-hmm.
On the surface of a cell, for example.
Yeah, so basically, did the changes that we made to the handle before, we wanted them to get around the virus stuff, but we still want that handle to work for other purposes, like turning on NK cells.
Yeah.
All right, so they take human peripheral blood monocytes, PBMCs, and they have NK cells in them. In that
Well, they're
population.
mononuclear cells, not monocytes.
Mononuclear cells. And then they incubate these with target cells that are producing GB, and they have the tool GB antibody in there, either wild type FC or with the YTE FC. Okay? And then they have a tool antibody with amino acid changes that completely eliminate effector function. That's a negative control. So then they look at ADCC-mediated killing in this assay, and they compare the tool YTE relative to wild type. They get a modest reduction in ADCC-mediated killing with the tool YTE relative to the wild type FC. So modest reduction, even though you've got these changes in the FC, but it still showed substantial killing above the effector dead change that they introduced into the FC. Right.
So you lost a little bit of NK activation, but you didn't get rid of it entirely.
Yeah. They also look in guinea pig splenocytes, because we're going to see in a moment, there is a nice guinea pig model of herpes simplex infection. These YTE changes only slightly reduce the ADCC of the tool antibody with guinea pig effector cells. So they say using these YTE engineered antibodies is a good way to avoid immune evasion by simplex, but to maintain the effector functions. That's the point here. You haven't destroyed the effector functions, which may be important for controlling herpes virus infections. Okay. Now the guinea pigs. So the guinea pig is really the only model for recurrent genital herpes. So you infect vaginally, you get acute disease for about 2 weeks, and then you get a latent phase where the disease can reactivate spontaneously, and you get lesions, right? So then they did 2 kinds of experiments to see if you can prevent acute disease. They have one where you give antibodies a day before infection, so that's prophylactically, and a second where you give antibodies 2 days post-infection. And they have a positive control. They have a drug, pritelivir, which is a helicase primase inhibitor, which will prevent disease. So for the prophylactic study, where they give the day before, the tool anti-GB antibody plus the drug PTV significantly reduce the lesion rate, so that's what the score is here, and reduce the viral load compared to untreated animals. So they do vaginal swabs and they look at viral load. So it reduces lesion rate and viral load, and that's in alignment with previous studies. They see a really good reduction in lesion rate when they use the tool and tool GD-GB bispecific antibodies when they initiate treatment 2 days before infection. They also use an effector dead antibody because we don't know the contribution of effector functions on this model. So they use an effector dead antibody and they get similar efficacy. So effector function is not required for preventing acute disease in this model. Okay, so looks pretty good at preventing acute disease. So what about if you initiate after latent infection is established? So the first one we're treating before the actual infection. Now we infect and let latency become established. So what they do here is they give antibody twice weekly from 20 to 40 days post-infection, because you don't know when the reactivation is going to occur. So you just dose them continuously, and you hope that in that time you're going to see some reactivation in your untreated controls. And so then they look at the lesion score of animals dosed or not dosed. So, gosh, when they have a whole page of figures,
Yeah.
no reduction. So then the sentence is split. No reduction. Scroll a page in lesion rate or cumulative mean lesion score observed in animals dosed with the tool antibody compared to the isotype control. Drug treatment significantly reduces the lesion rate and lesion score. So the efficacy of a CS blocking antibody can't be inferred by its ability to block acute infection. So it's not working in this reactivation model.
And that's for the tool.
That's for the
tool.
tool antibody. Which prevented lesions after the initial
In the
infection
acute
quite
model.
well.
Right.
Right, in the acute model. Okay, so then they look at their bispecific antibodies in this model. This is the experiment we're really waiting for, right? So they have humanized bispecific antibodies. They have D7, D1, where GD arm is first, and then they have another one where the GB arm is first. They have the FC modification, and then they use those in the guinea pig models. Now, this FC change is in the human FC portion here. It's all human, and these are guinea pigs. And so they say we don't expect it to increase the half-life in guinea pigs, and in fact, it doesn't. So that's why they did a twice weekly dosing for a long time, because normally in humans it would last theoretically a long time, but it's not in guinea pigs.
Because the guinea pigs are making a response to this foreign immunoglobulin, which humans would not do.
So when you dose from 20 to 40 days, both of these antibodies significantly reduce the lesion rate, as well as the cumulative lesion score compared to untreated animals. There were zero lesion or close to zero lesion recurrences after the third dose of both bispecific antibodies. But in contrast to what we see in humans, adding the antiviral only has a moderate effect on shedding and no effect on viral DNA copy in the swabs. So they think the guinea pigs are actually underpredicting the clinical efficacy of these antibodies.
Which would make sense given the half-life difference.
Yeah.
And finally, they look at serum levels of these antibodies after the last antibody dose. They're about 1 mg per mil, which is about what's been achieved in previous studies. So this is a nice proof of concept that this anti-GD-GB bispecific antibody, which is really good at neutralizing virus and blocking cell-to-cell spread, suppresses recurrent genital herpes. That's cool.
Yeah.
That could be a big help for a lot of people.
Yeah.
Yeah, so what's the therapeutic modality here?
They suggest using an mRNA delivery. So instead of manufacturing the nanobodies and delivering them that way, deliver them as mRNA and have them made in the cells.
Interesting. So you would...
Which gets you around a manufacturing bottleneck.
Right.
Well, this would have to be given multiple times.
Periodically, yes. And
periodically.
whatever. They would need to do a human trial to see what the half-life of this thing is in humans, because it's a humanized antibody, and then base the frequency of dosing on that.
Right. I mean, you could go with the protein in the way that they do with some of the anti-cytokine antibodies for autoimmune disease.
Yeah.
But I think that you're completely right that going with the mRNA delivery of a gene encoding this modified antibody makes more sense.
What about for prophylaxis? Could that be used as well? It's harder because you don't want to be prophylacting all the time, right?
Right. Yeah, I mean, I guess, you'd have to.
I mean, you could, if you're in a high risk group, and depending on what the dosage schedule is, if this is a lenacapavir type of twice a year thing,
Yeah, that would
might
make
be
it
worth
easy,
it.
but
Yeah.
I don't know if it's going to be. That could be every couple of months, depending on how
Yeah.
they modify it further. Okay. Anyway, they talk about, so there's, as we said earlier, there's no vaccine. And there was a recent evaluation by GlaxoSmithKline of a vaccine for genital herpes. It failed and it's been discontinued. So there's nothing in the pipeline. So this is, they say the potential roadmap for the design of mRNA encoded passive immunotherapy. Yeah. That would be interesting. But this is going to be difficult in this climate in this country, right?
Right.
Sure.
These people at HHS some people don't like mRNA vaccines, and it's unfortunate because there's nothing wrong with them, and they can be used for other things like this too. So to put a damper on their development is completely uncalled for, I think.
Yeah.
Based on no science whatsoever. Anyway, I thought that was very interesting. The way they made these antibodies, the logic, and how they work very well. Beautiful done. Nice. Very nice. Very nice. Okay. Let's do some email. Brianne, can you take the first one, please?
Sure. Rebecca writes, "Hi, everyone. I just listened to TWiV 1243 and got a bit starry-eyed when I heard my name mentioned. Thank you so much for the lengthy discussion of the Nature paper." This was the previous episode where we talked about the paper that had made a bit of news about respiratory infections and breast cancer recurrence. As a cancer survivor, uterine, and 13 years post-treatment, it was gratifying to hear you explain some of the mechanisms that this paper describes, and I was particularly interested in the open questions that everyone identified. I definitely wish I had a deeper science education than I do. My key take-homes are these two things. I'm going to be much more assiduous about masking. The primary metastatic site for uterine cancer is lungs, and I am going to decline nasal administration of vaccines. I'm 70, generally in very good health, and I would like to keep it that way. Thanks again. Warmly, Rebecca. Rebecca's from Seattle, and Rebecca says, P.S. I remain particularly frosted about the idiot decision not to offer COVID vaccines to people under 65. My husband is under 65, and that puts me at higher risk. I am not the only one in that position. Mr. Kennedy does a good job of presenting as a eugenicist. Onward.
Yes, I like that. Frosted.
Yes.
That's really good. Yeah, I think that's good logic. We don't know about other cancers, but if the primary metastatic site is in the lungs, then it's possible that they make dormant cells. Yeah.
Yeah, that makes a lot of sense to me.
And a couple of straightforward interventions like, just taking masking more seriously and trying to reduce your exposures. Sure.
Alan, can you take the next one?
Sure. Jane writes about the Tour de France stage 19, which was rerouted to avoid diseased cows. I think we talked about this previously. Quote, contagious nodular dermatitis, lumpy skin affecting cattle in a herd located specifically in the Col de Cézes. Did I get that right? Cézes necessitated the culling of the animals. Maybe Angela could give us insight into the disease in cows. How did the cows get it? Another mammal? How sick do they get? And humans? A milk the cows stop for the riders? Seems unlikely. However, the Tour de France is unique in that many people, children and their pets, are lining up along the route, and there are huge numbers of spectators. That brings many humans into the countryside for hours or even overnight, waiting for the riders to go by. Maybe the distressed farmers are just not up for all the crowds around their farms this year. Can't say I blame them. Could the source of the virus be a different mammal? And Vincent has a note here. Nodular dermatitis in cattle is caused by lumpy skin disease virus, LSD virus. It's a capripox virus, mainly affecting cattle, and newly emerging disease in some parts of the world.
But really importantly, it is transmitted primarily through arthropod bites.
Yeah.
And so those spectators, one might imagine, could get bitten by an arthropod that had previously bitten one of these cows, and the cows probably got it from being bitten by a biting fly, mosquito, or ticks. Although, again, as Vincent pointed out here, direct contact can play a role too.
Yeah.
I have a picture in the article of some cyclists cycling past cows. Huh. I don't. Yeah, I guess they're just worried about transmission. Oh, they culled the animals, so maybe they're culling activities too, and that's not nice to look at.
No.
That's too bad. Poor cows. I'm sorry, cows. Okay, Eileen writes, not jackasses, y'all. Very interesting development of research. Donkeys against deer ticks. Let's hope their funding continues. Fascinating. So this is a article from the University of Massachusetts Amherst.
Yeah.
CNS researchers find that a donkey skin secretion naturally repels deer ticks. Donkeys are very cute, especially small. Do you ever see a tiny donkey? They're so cute.
Yeah.
They found that a naturally occurring compound repels Ixodes scapularis ticks, common tick in the U.S., the deer tick. They have a little assay here showing the inhibition of their movement. Well, they have to isolate the active ingredients, right? You're not going to just grind up donkey skin and give it to people. So we're going to find out what it is and synthesize it. So that's cool.
Nice.
On the eastern edge of Western Mass.
Yes.
So it's not far from you, right?
Yeah, that's got to be, I'm guessing, out somewhere near Worcester.
Brienne.
Donna writes, you guys do crystallography. How can you say there are no right angles in nature? I'm a retired teacher who dabbles in geology, and I can assure you there are right angles in nature, maybe even in biology, although I'm not sure about that. Keep on fighting for science.
So this came up during, I forgot, a couple of TWiVs ago. Does anybody remember what this was about?
I remember something about this.
We were saying that something was artificial because of right angles.
No. I think, I think they're right. I mean, crystals have right angles, right?
Yeah.
Mm-hmm.
And Kathy was here talking about it. I can't remember what side she was on. Maybe she's the one who said that someone said there are no right angles in nature. Yeah, I don't remember. I think there are. I think you have to be careful about no statements.
Yeah.
Right? Alan?
Okay. Marshall writes, if I might, let me offer my personal idiosyncratic definition of science. A collection of techniques intended to minimize self-deception. Thank you for all that you do to stand up for science and the pursuit of truth. I like that.
That's good.
And I can go ahead and take Chaim.
Yeah, do the next one too.
One-sentence email. Chaim writes, on the topic of footnotes and references, the science journals still title the section as References and Notes, with the author guide stating, notes should be used for information aimed at the specialist, e.g. procedures, or to provide definitions or further information for the general reader that are not essential to the data or arguments. It does seem that they are rarely used, but here is a not-so-old example that I happen to be deeply familiar with, provides a link, PDF attached. As long as I'm writing a listener pick. Shall we do this one for this episode?
Yeah, sure.
Sure. I'm currently reading Virus Hunters by Greer Williams, published in 1959. It's a history of the field of virology and vaccinology from Jenner to the beginning of clinical trials by Enders and Peebles, with a final section on DNA and the then still controversial possibility of viruses causing human cancer. The science has come a long way in 65 years, of course, but the book is focused more on the personalities and the history. Entertaining and informative. Thank you.
Could you copy that and put
I'm putting it down with
the
listener picks.
Michael writes, This is a wonderful interview with Meselson and Stahl. It includes a history of the experiment and many great stories leading up to and following the publication of the results. Wonderful, enlightening, and fun. And Michael provides a link to that YouTube video. And I'll also take the last one. Bridget writes, I've been a TWiV listener since December 2020. At that time, I was looking for more science-based pandemic information, and TWiV was exactly what I needed. I think it was a tweet from David States that first introduced me to TWiV. I've listened to every episode except for 21 through 254. Even though I'm not a virologist, I felt like I found a group of like-minded people to listen to multiple times a week. I am just, in quotes, a mathematician who happened to get her PhD at Carleton University in Ottawa, Ontario, and years ago I co-taught a calculus class with Angela's dad. How about that? I was so happy to hear alpha-1 antitrypsin deficiency was mentioned multiple times on TWiV. I accidentally discovered that I have this deficiency when I did 23andMe over a decade ago. Luckily, I'm not symptomatic, and I'm currently participating in a clinical trial. This leads me to my listener pick, a biotechnology company called Wave Life Sciences, and their potential treatment for alpha-1 antitrypsin deficiency called WVE6. WV6 is a GalNAc conjugated, subcutaneously delivered A to I RNA editing oligonucleotide. It's currently in phase 1b/2a clinical trial. WVE6 corrects the error in the mRNA so that normal alpha-1 antitrypsin is made instead of the misfolded protein. If this treatment works, it may potentially prevent the lung and liver disease that is seen in people with alpha-1. Early results published in the fall show promise, and Wave Life Sciences shared this update in October 2024. Quote, today's proof of mechanism data are the first ever clinical demonstration of RNA editing in humans. These data are from the first single dose cohort, 200 milligrams, in Restoration 2, and include the first two patients with ZZ AATD to reach day 57. Individuals with PI ZZ AATD do not naturally produce wild-type alpha-1 antitrypsin protein. Therefore, the presence of M-AAT protein is confirmation of successful editing of mutant mRNA. Additionally, restoring 50% M-AAT would be consistent with the heterozygous MZ genotype with low risk of lung and liver disease. And there's a link for that. I'm in the double dose cohort, and I've recently completed the multi-dose portion of the study, a dose every 2 weeks for a total of 7. Wave Life is expecting to share more results sometime in the third quarter of 2025. They also have clinical trials for Huntington's and Duchenne muscular dystrophy that are showing good results. Thank you to all of you for your wonderful podcast. Bridget is in Gatineau, Quebec, Canada. Now, let me see. I'm trying to figure out RNA editing. So what are they delivering? They're delivering an oligonucleotide. But how's the editing being done? Does anyone know?
I don't know.
I have an idea, but
Therapeutic RNA editing. Of course, reading the press release is not helping because there's not enough.
Yeah, I went to the technology section. There's PRISM RNA Medicines platform. I'm looking at it now. And it's all about how wonderful it is, but it's not giving me. I'm sure it's in here somewhere.
Interesting. Well, thank you, Bridget, for that. It's cool that you're in the clinical trial.
Yeah.
I like that. All right. So, but, you got... you don't know if you've got the placebo or not, probably. And we don't know how they're going to assess disease. I guess it's just the presence of the protein. Yeah. All right. It's time for some picks. Sorry, go ahead.
I'm not sure exactly all of the details, but it does involve ADAR and double-stranded RNA. So in theory, they're delivering some kind of oligo that would either lead to double-stranded RNA production and then the editing of the mRNA, or that would then, they are delivering double-stranded RNA that somehow then gets edited.
Yeah. Interesting. Time. We had 2 listener picks already, so that's
Yeah.
great. Thank you so much. Time for our picks. Brianne, what do you have for
Um,
us?
I have an article about a study, and I've seen this come up in a few different places. It is reporting on a study that came out in Lancet, Gastroenterology and Hepatology. And it is actually looking at AI use in doctors. And because I've been doing a lot of course prep and I've actually been in multiple meetings lately where we've been talking about AI use and things, this definitely was particularly of interest because this study showed that doctors who were using AI to look at colonoscopy data actually became less skilled at cancer detection of those colonoscopies. So basically, these physicians were using AI to try to help them detect the tumors and colonoscopies. And the physicians who frequently were using AI actually became less skilled because of their reliance on AI. They sort of lost the skill because they weren't practicing it as frequently.
Yeah.
And so I thought that was a really interesting thing that we should think about in terms of, yes, there are things that we can have AI do for us. I was sort of at first interested just because of the idea of, no, there's some skills we have to have and we have to practice in order to learn them. But I was actually very surprised that in the study, they actually mentioned that those who were very clinicians became over-reliant on AI recommendations and became less motivated, less focused, and less responsible when making cognitive decisions without AI assistance. So my first thought about all of this was just about it's important to practice a skill and keep it sharp. Yeah. But the idea that there might be changes in motivation or focus or something like that is even more worrisome. They do point out in this article, which I linked from Time, as well as in some other places, and even in the discussion section of the paper, that the study design might have overestimated this decrease. And so maybe it's not quite as bad as they indicate here. But I think it's something that we should certainly be aware of in thinking about AI use.
Yeah.
So in a colonoscopy, they take a video of the camera going through the colon, right?
Mm-hmm.
And then, I mean, I was awake for one once, and you say, "Oh, there's a polyp," So they could either see it on the first shot, or they can go back and look at the video, right?
Mm-hmm.
And say, "Okay, you have this." And sometimes they biopsy it right there, right? So I don't know where AI would even come in and
highlighting what's a polyp, maybe?
Yeah, but during the procedure?
I don't know.
I don't know. I couldn't figure
I don't know
that
how
out.
it's implemented.
Because during the procedure, if they find a polyp, typically they'll
biopsy it, right?
Biopsy it. If they can pull it off with a wire, they will do that as well. Yeah, because you want to know. And then and typically, unless you're in an advanced state, there's one. So I don't know where the AI would be. Maybe later watching the video. The AI.
That's kind of what I am imagining.
And then the physicians would be like, okay, that's it, and not look anywhere on their own. Is that the idea? Maybe?
I think that's the idea, is that they basically let AI look or let the physicians look. And the physicians who had previously been using AI got worse.
Well, did the AI get better?
Probably not.
It doesn't seem like it.
This is very tough.
Yeah. It's kind of like how everybody forgets everything now that we have Google.
Yeah.
Yeah, well, at least you know where to look for it, Alan.
Yeah,
right?
it's not. This is a more severe version of the problem.
Procedures that physicians do are good if you do a lot of them.
Right.
Right.
Yeah.
And so that includes looking at the results. So, I mean, I don't know what the aim is for using AI here, but it just bothers me,
I think that the overall idea is that, in theory, if AI is really well trained, AI could be so much better. And perhaps, you wouldn't have to have so many highly trained doctors, because you could
expensive, yes.
Yes, highly trained, expensive doctors. You would just have this great AI to do this very cheaply. But this is a real downside. of
Yes.
that.
Well, but you got to still have the procedure done. AI is not going to scope you.
True.
Right.
I hope not.
So a robot, Dr. Dove, it's time for your
Yeah.
call.
No.
So these expensive doctors are expensive because they're highly trained. And,
Yes.
they're going in your body and you want them to have steady hands and to have done it 50,000 times,
Yeah.
right?
Mhm.
So they're going to get it. So it's like second nature. And
Yeah.
I don't know how AI is going to.
Yeah.
I mean,
what-
I think it's very interesting. Don't get me wrong, but I'm trying to figure out what they're trying to do here.
In some educational discussion or reading that I did earlier this summer, I heard an analogy that I really stuck with a lot, and I feel like this article played into my new favorite analogy, which is that sometimes AI is like using a forklift at the gym. Like
Yes.
like the idea is that sometimes, yes, it can help you lift the weights, but sometimes the point was actually you getting something out of lifting
Yes.
the weights. And it would be silly to have some AI do it for you.
Yeah.
And so here it's sort of showing the point of sometimes you are getting something out of doing this process yourself.
Yeah.
I mean, you think, not today, but, maybe in 100 years, people will be accepting robots doing their robotic surgery without any humans involved.
In science fiction books, they say yes, but I don't know.
Yeah. It would have to be a generation after me because I'm not going for that.
Yeah, 100 years.
Yeah.
We're not going to be around.
No,
but,
that's fine. That's going to be somebody else's problem. But
it will slowly insinuate, and then there'll be clinics that do it, and it's very inexpensive, and people will start to go there, and then it will permeate, how it works, right? Very interesting times we are in, always.
yes.
It's very cool. Alan, what do you have for us?
I have an article from Science. It's a news article by John Cohen about a meeting that occurred, how recent was this? Back in May, of conservation biologists and microbiologists about basically save the microbes. So what they're advocating is there are rare, potentially endangered microbes usually in unusual environments, whether it's hot springs or deep sea vents or high salt lagoons, and we really should take a serious look at establishing some conservation practices the way we do for macroscopic species. You say you shouldn't cut down this particular area of forest because there's this rare owl that lives there. Well, maybe you shouldn't fill in these salt-rich lagoons because there's this rare bacterium that lives there. And I think it's a really neat idea to say that there are microbes that are worthy of conservation. These are species that we want to keep around. They're doing something in the environment. We may be very sorry if we get rid of them. And I think it's a cool approach, and they're going to try and pattern it on the IUCN's red list of endangered species, which uses designating an area that's a habitat.
I think this is a great idea. I really like the idea of thinking about keeping microbes that have important ecological roles around, and also thinking about keeping diverse microbes around, because who knows where the next tack or the next antibiotic or what have you will come from.
Yeah.
I like that this talks about the publicity problem, because I'm imagining trying to tell
Yes.
my non-science friends about saving the microbes, and I can't see them being super excited.
Yeah.
But my biggest question is, how do they know which ones are endangered? I feel like when you are thinking about these rare, not well understood organisms, how are they actually determining what endangered is for these organisms?
I think in this case, you'd probably be looking at the environment the organism lives in and how common is that environment on Earth. So deep sea vents are an obvious choice. You probably don't want to, well, there are a number of reasons you probably don't want to mess those up, not least of which is that they're a volcanic seam and you shouldn't be poking around it too much. But you shouldn't be doing mining or heavy industry there, because if you run out of those, you're going to run out of the whole ecosystem that's there. And some of these seem to be place specific. Nobody's isolated them in environments like that, or environments like that don't exist elsewhere because they don't have the same temperature or the same salinity or what have you. So I think it would just be on the basis of what does this thing need to survive and how likely is it that it's anywhere else?
Interesting. Also, we don't want to make bacteria do too much work for us.
No.
We have to be sensitive.
We have to, yes, because then we'll lose the ability to do our own work.
Okay, my pick is an article in Science-Based Medicine by Jonathan Howard. It's called Dr. Vinay Prasad's Catch-22. The only way he can truly succeed at his job is to get fired again. So, Dr. Prasad let me read a little bit, has become a lightning rod during his brief time at FDA. The drama has never been higher and trust has never been lower. Back when he was auditioning for his job, Prasad debased himself by relentlessly fluffing RFK Jr., his future boss, claiming he was more reliable than vaccine expert Dr. Peter Hotez, and that he would be the savior of vaccines. Dwell on that for a moment. In order to gain power, Dr. Prasad was willing to write this article called Sabotaging RFK Jr.'s Confirmation Will Increase Vaccine Hesitancy. Oh my gosh. Anyway, it's a very interesting article that talks about how the only way Prasad can succeed is to be fired because he's completely unqualified for his job. He has nothing to say as his boss spreads gross anti-vaccine disinformation and dismantles research in vaccines, even though RFK Jr.'s disgusting rhetoric inspired fatal violence at CDC and RFK Jr. continues to dunk on the people who work there. Prasad maintains his monk-like science. I like this one, too. RFK Jr. is the literal chief of the medical establishment and is a significant threat to the health of American children. However, Dr. Prasad is mute. So anyway, it goes on and on about that. And it says basically he should quit and then we won't be subject to his disgraceful behavior. Very interesting column. I think it's too bad he came back. It just illustrates how this administration doesn't really do its work.
Yep.
You know, some right-wing columnist trashes him, Prasad, and they say, okay, he's got to go without doing any research. Not that they should have brought him back. They should have gotten rid of him to begin with, but not for the reason that Sheep said. It's just crazy. It's disgusting how no expertise is apparent in this regime.
Not even no expertise, unexpertise.
Yes. There's an
Unexpertise, the opposite.
anti-expertise bias.
Unbelievable. And we also have picks, of course, which you've heard already by Chaim and Bridget. That is TWiV 1245. You can find show notes at microbe.tv/TWiV. You can send us questions, comments, picks of the week to TWiV at microbe.tv. And if you enjoy this work, we would love to have your support. Microbe.tv/contribute. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv or microbe TV without the dot on Blue Sky. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene Ramsey for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.